Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction, and it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next. Propagation of action potential in an unmyelinated axon. In unmyelinated axons, action potentials propagate as an interaction between passively spreading membrane depolarization and voltage-gated sodium channels. When one segment of cell membrane is depolarized enough to open its voltage-gated sodium channels, sodium enters the cell by facilitated diffusion. Once inside, positively charged sodium ions nudge adjacent ions down the axon by electrostatic repulsion and attract negative ions away from the adjacent membrane. As a result, a wave of positivity moves down the axon without any individual ion moving very far. Once the adjacent segment of membrane is depolarized, the voltage-gated sodium channels in that segment open, regenerating the cycle. The process repeats itself down the length of the axon with an action potential regenerated at each segment of membrane.